Hello viewers, welcome to my channel Biology Notes. Today I am going to discuss about T lymphocytes or T cells. The letter T stands for thymus, the maturation site of these cells. In this chart, you can see T lymphocytes are divided into two major cell types, T helper or Th cells and T cytotoxic or Tc cells. T helper cells and T cytotoxic cells can be distinguished from one another by the presence of either CD4 or CD8 membrane glycoproteins on their surfaces. Th cells display CD4 on their membrane and recognize antigen in complex with MHC class 2. Tc cells display CD8 on their membrane and recognize antigen in complex with MHC class 1. The ratio of CD4 plus to CD8 plus T cells is approximately 2 is to 1 in normal human peripheral blood. A change in the ratio is often an indicator of immunodeficiency disease, autoimmune diseases, and other disorders. Now, these two major cell types can be differentiated into some other subsets when activated. T cytotoxic or Tc cells differentiate into effector cells called as cytotoxic T lymphocytes or CTLS. T helper or Th cells can differentiate into a variety of effector T cell subsets. T helper type 1 cells or Th1, T helper type 2 cells or Th2, T helper type 17 cells or Th17, T follicular helper cells or TFH, regulatory T cell or TREG, etc. Now I will discuss the functions of the T cells. Suppose this is a naive CD8 plus T cell. The naive CD8 plus T cells browse the surfaces of antigen presenting cells with their T cell receptors. If and when they bind to an MHC1 peptide complex, they become a cytotoxic T lymphocyte or CTL. The peptides or foreign antigens may be virus infected cells, tumor cells, or cells of foreign tissue graft. Mature cytotoxic T cell now can kill cancer cells, cells that are infected by intracellular pathogens, or cells that are damaged in other ways. To proliferate and differentiate optimally, the naive CD8 plus T cells also need help from mature CD4 plus T cells. Suppose this is a naive CD4 plus T cell. The naive CD4 T cells also browse the surfaces of antigen presenting cells with their T cell receptors. If and when they recognize an MHC class 2 peptide complex, they can become activated and proliferate and differentiate into one of a variety of effector T cell subsets. When they differentiate into T helper type 1 or Th1 cells, then Th1 cells regulate the immune response to intracellular pathogens. When they differentiate into T helper type 2 or Th2 cells, then Th2 cells regulate the immune response to extracellular pathogens. When they differentiate into T helper type 17 or Th17 cells, then Th17 cells secrete IL-17, which plays an important role in cell-mediated immunity and may help the defense against fungi. When they differentiate into T follicular helper cells or TFH, then TFH cells play an important role in humoral immunity and regulate B cell development in germinal centers. When they differentiate into regulatory T cell or TREG, then regulatory T cell has the unique capacity to inhibit an immune response. These cells can arise during maturation in the thymus from autoreactive cells called natural regulatory T cells, but also can be induced at the site of an immune response in an antigen dependent manner called induced regulatory T cells. Regulatory T cells are critical in helping us to quell autoreactive responses that have not been avoided via other mechanisms. Regulatory T cells also play a role in limiting our normal T cell response to a pathogen. So, this is all about different types of T lymphocytes and their functions. If you have any query, please comment. Subscribe the channel for more new videos. Stay with me, keep watching. Thank you.